Assalamualaikum and good morning, and welcome to this morning's episode of Breakfast at UMHealth. I'm Azlina and I will be moderating the first session. Let me introduce to you the first speaker. Her name is Associate Professor Dr. Puteri Shafinaz Abdul Rahman from the Department of Molecular Medicine. Dr. Puteri is a alumni, having received her undergraduate education in this university. She then pursued her PhD at the Nottingham Trent University, where she was awarded the Commonwealth Scholarship and the Fellowship Commission, United Kingdom. She trained as a molecular biologist and her research mainly focused on proteomics. Dr. Puteri was previously appointed as the Deputy Dean of the Innovative Technology Research Cluster, as well as the Frontiers of the Natural World Research Cluster. She currently heads the Central Research Laboratories, as well as the Center for Proteomics Research, a center that she co-founded with another researcher. So this morning, she's going to share with us some of her research that she has done on the title of Dynamic Changes in Protein Composition of Human Milk, from Tiny Tots to Toddlers. The floor is yours, Dr. Puteri.
Right, thank you very much, Dr. Azlina. Okay, let me just switch on to my presentation. Okay, are you able to see my screen right now, Dr. Azlina? Prof. Azlina?
Yes.
All right, okay, thank you very much. So, Assalamualaikum and a very good morning to everyone who's present today. And thank you very much, Prof. Azlina, for the kind introduction. I'm happy to actually be here this morning to share a little bit about the work that my team has been conducting in our lab, which is entitled Dynamic Changes in Protein Composition of Human Milk, from Tiny Tots to Toddler. Yeah. So maybe just a bit of background in terms of my work. I'm actually a proteomics scientist. Earlier part of my career, I have actually been working or focusing on cancer biomarkers, looking at the potential biomarkers that can be used for early detection of cancer. My work also focuses on natural products and use of proteomics technology to actually understand the mechanistic basis of the compounds and how they can actually be utilized for treatment. Okay, so in terms of human milk, this is considered quite a recent project in my team. Okay, we've done it for about 8 years, yeah, and out of that, we've managed to publish a few papers, and I think the interest has really grown within our team to sort of like look into milk biology in depth. Okay, so without further ado, let me go over the overview of my presentation. Okay, I'll start with an introduction, just a short introduction in terms of milk composition, what are their functions, and also comparison with formula milk and the breastfeeding status in Malaysia. Okay, following that, we move to the lab where we look at the research part of human milk proteomics, and finally, it'll be a summary of our findings so far. Okay, so as an introduction, okay, human milk is actually a very important source of nutrient for infant growth, especially so if we talk about the first 6 months of life. Okay, and human milk has been sort of like described as a living fluid as it changes from day to night and throughout the entire breastfeeding period. So studies have shown that composition of milk is different at day and also at night, whereby there are different hormones that are released, or also in terms of nucleotides. Okay, for example, at night, it's higher in content of hormones and also nucleotide, which helps in sleep regulation for the babies. Yeah, and when you say throughout the entire breastfeeding period, it would be the milk actually changes its content according to the different stages of development, or let's say as the child grows. Okay, so substantial amount of research on lactation, human lactation, has been performed, okay, to understand how human milk is adapted to the infant's metabolic, digestive, physical development, and also the child's defense requirement. Okay, and studies also focus on the nutritional immunological advantages, prevention of allergies, intellectual development, mother and child bonding, as well as psychosocial effects of breastfeeding. So why is it that, relating to the title of my presentation, whereby we focus from tiny tots to toddler, why is it that we want to look at this longer time frame of breastfeeding? Okay, it's because it's been acknowledged that the time between birth and 2 years old is actually the critical window for the child's growth and development. And if you look at sort of like the growth of brain, it is actually during that time that it grows considerably, whereby it's actually the second year of life, and therefore the components of human milk, such as the protein, fats, and vitamins, they are the crucial components towards development of a good connective for good connective development. Yeah, and studies have shown that long-term breastfeeding will create a foundation that is just not during infancy, but also it leads to a good health and well-being well beyond later years, as the children grows to be adolescent, and also when they grow up as adults. Okay, so when we actually looked at current literature available, we saw that there is actually a lacking in terms of description for components or composition of human milk. Most studies actually look at studies for milk which are under 1 year old, or mainly more of the literature focus on prior to 6 months. Okay, so this is where we were interested to actually look at human milk composition in extended breastfeeding. Right. So having said that, maybe I could elaborate a little bit in terms of the composition of human milk. Yeah. So this picture here shows that, breast milk is a living tissue. So even though it's made up of mainly water, 90% of it, okay, it's still within it contains nutrients such as fats, carbohydrate, proteins. Okay, these are the macro nutrients, and also the micronutrients such as vitamins and minerals. So that makes up sort of like the nutritive compartment of milk, whereas there's still factors which are bioactive that are present in human milk. This would be the immunoglobulins, and as I said earlier, hormones. You have human milk oligosaccharides, antimicrobial peptides, and microRNAs, and so on. Also, in terms of in milk itself, there are commensal bacteria. So all this breast milk component actually also helps. Also, these are the component that exert its effect for infant development, okay, and play numerous physiological activities, which includes in terms of the development of immune function, defense against pathogenic bacteria and viruses, and also in the development of gut. So having described to you about the composition of human milk and what benefits it can actually provide to the child, okay, you may wonder how much difference is there in terms of components of human milk compared to formula milk. Yeah, some babies are also given formula milk. So in this poster, you could actually see, okay, this is a poster by the NHS in Scotland, okay, to show the comparison between formula and also breast milk. Yeah, it states here that breast milk contains 34 important ingredients not found in infant formula. Okay, but recent studies have shown there's actually much more. Okay, the components that are found in breast milk, it's probably 2 to 3 times much more compared to things which are available in formula. And if you see right now, okay, for formula, it is mainly the sort of like nutrition content, which is protein, water, fat, carbohydrates, and the vitamins and minerals. So this next slide also shows in terms of when you look at different composition of human milk, cow's milk and formula. So bear in mind that in the market right now, most of the formula are actually derived from animal milk, which majority of it is actually from cow's milk and also from goat's milk. Okay, but you can see in terms of the pattern or percentage distribution, okay, of the macronutrient here, it is not similar, okay, for human milk, cow's milk, or formula. For human milk, about 54% of it, the distribution actually comes from lactose. And if you look at this purple bar, this is the oligosaccharides, yeah, and you can see it's very different from cow's milk. You don't see much of oligosaccharides, and the proportion of, or the percentage, is about 1/3 each for lipid, lactose, as well as protein. Compared to formula, on the other hand, okay, higher percentage of lipid component, okay, so these are also in terms of, when I mentioned just now, let's say formula is from derived from cow's milk, so these are not just sort of like lipids from cow milk itself, but also manufacturers tend to fortify milk with more added fats, usually from plant products. Yeah, so just to if we focus on the human milk oligosaccharides, okay, just to show, this is a screenshot of quite a recent paper by Lars Boot. He's actually a renowned milk biologist, okay, entitled Human Milk Oligosaccharides. Every baby needs a sugar mama, okay, because the oligosaccharides that is mentioned here as HMO is actually sugar molecules that are found in milk. So the oligosaccharides, when through research, they found that they are a family of very structurally diverse glycans, or in layman term, it's B sugars, that are highly abundant, and it is unique to human milk. Okay, and there's also evidence to show that the HMO actually serve as soluble decoy and help to prevent pathogen attachment to the mucosal surface, and therefore it can actually lower the risk for viral, protozoan, and also bacterial infections. And in addition, it's found that the HMOs may modulate immune cell responses and lower the risk for necrotizing enterocolitis. And other papers have also shown that the HMOs are rich in sialic acid, which are essential nutrient for the brain development of infants. Yeah, so therefore this further shows the evidence of the superiority of human milk, and how that human milk is specific and designed for human babies. Okay, so I just like to share some quotes that I found, which relates to the presentation that I'm giving today. Okay, if you look at the one in purple, mother's milk developed through evolution with thousands of ingredients to build the human brain, body, and immune system, is incomparable with man-made product of a couple dozen ingredients that are non-human and in the wrong proportions. Okay, and another one says, something as simple as better breastfeeding could save 1 million children a year. Okay, so having said that, we look at the breastfeeding in Malaysia in terms of how the scenario is in our own country. Okay, we have a national performance key index that targets for exclusive breastfeeding for up to 6 months, which is at 58%. Okay, but a study which was done by the Institute of Public Health in 2016, okay, in their survey for the National Health Mobility, showed that the achievement for exclusive breastfeeding for up to 6 months is 47.1%. Okay, and when they actually look at the prevalence of continued breastfeeding among children aged 20 to 23 months, the percentage is even lower. It is at 39.4%. Okay, so this could actually sort of like be due to the challenges that are faced by mothers in Malaysia trying to breastfeed for a longer period of time. Okay, in Malaysia, most of the mothers are actually working mothers, and of course, when you work in terms of trying to sort of like breastfeed your child for a longer period of time, or even providing express breast milk, can be a challenge. Okay, and of course, there also need to be support from family members and to motivate the mothers to continue breastfeeding longer. And there also is lack of knowledge in terms of for mothers. Probably they do not know that longer breastfeeding period actually gives better health for their children. Okay, and following that, in Malaysia, the government has actually crafted the National Plan for Action for Nutrition. This is for spanning the year of 2016 to 2025, whereby the target is to have 100% breastfeeding initiation within the first hour of birth, and the target for exclusive breastfeeding for the first 6 months of life is raised to 70%. Okay, and to do this, they are trying to promote action and improve nutrition for mothers and children in the first 1,000 days. Okay, I also came across this sort of like research that's done by a global NGO, Alive and Thrive. Okay, so they published this sort of like their research in this Health and Policy Planning Journal, which is quite recent, 2 years back in 2019. And what they looked at is that they quantified the economic cost of not breastfeeding on human capital development and health systems in Malaysia. So this is actually a part of their breastfeeding advocacy series, whereby they actually looked at breastfeeding costs in a few countries such as Malaysia, Vietnam, Cambodia, South Africa. So I found this for Malaysia. Okay, so in short, these are their key findings for the cost of not breastfeeding in Malaysia. Okay, so they find that each year, if we actually have optimal breastfeeding practices, then we could have the potential to prevent child death, okay, especially for under 5-year-olds, prevent maternal death from cancers and type 1 diabetes. Okay, we could have savings of about 27.8 million in healthcare system treatment, and we could also generate an additional 5.4 billion for the economy. Okay, this is actually looking at, let's say, in terms of child's productive years, by increasing the cognitive capacity and presenting premature mortality in the early years. Okay, and last and not least would be to reduce families' pocket expenditure. Of course, when you actually rely on formula, the costs are quite significant and can actually reduce the family's disposable income. Not to mention as well, sometimes you have this tendency to have sort of like these sickness and things like that for babies who are not breastfed, right? So moving on to the research part of the work. Okay, as I mentioned just now, we looked at available literature and we found that the studies for protein composition during long-term breastfeeding are still lacking, and we feel that it deserves attention. So therefore, our current study here actually is aimed to uncover dynamic changes in protein composition of human milk over time. And subsequently, we would like to elucidate what are these proteins' respective functions in infants' growth. Okay, in terms of methodology, okay, we actually analyzed samples of human milk from mothers that is that we collected around Klang Valley. Okay, these samples were obtained at different time points in the order of once every 3 months up to the 24th months. Okay, I have actually highlighted here in bold. This would be sort of like the samples that we have managed to analyze up to date, okay, and still pending for 18, 21, and 24th months. A bit of our sample collection were also delayed due to MCO because of the restriction of crossing borders and things. We were not able to collect the milk samples from mothers. We've only sort of like managed to collect some of it now, so we are still pending for 18, 21, and 24 months. Yeah. So for sample processing, we actually centrifuge the sample to separate the cream layer and the soluble fraction. Okay, for this part of work, we actually look into the soluble fraction. So that is actually equivalent to whey when you talk about animal milk. And following that, we sort of like process the sample for digestion. Okay, this is using enzymatic reaction such as trypsin to break down the proteins to smaller molecules or peptides that can be analyzed using proteomics analysis. Okay, so for those who are not familiar with proteomics analysis, this is actually a study that the technology actually enables us to profile complex mixtures of proteins simultaneously using various type of platforms. Okay, one of it is gel-based approach, but the one that is in choice for this particular analysis, we are actually using a mass spectrometry instrument. Okay, and from there, the data that we obtained from our mass spec analysis was analyzed using software such as Max1 and also Perseus. Okay, so through the information we collected from the participants, we formed this demographic data. Okay, the categorized based on maternal age, ethnicity, employment, education, and gender of infant. Okay, so it is important to note that, as I mentioned just now, our study is actually ongoing. So our aim is actually to continue collecting milk samples till the 24th month. And as a frame of reference, the recruitment of these women actually began in 2020. Okay, this is part of the data that we obtained. First figure here shows concentration of proteins in different months of lactation. Okay, so this graph actually shows that the concentration of proteins is generally stable throughout the lactation period of 15 months. Okay, however, when we look at the next figure here, figure 2, in terms of number of identified proteins in human milk for each time point, this is actually sort of like derived from our mass spec analysis. Okay, you can see that sort of like the lowest number of proteins detected is at the 3rd month, and it sort of like goes up quite dramatically in the 6th month. Following that, it remains stable for the following time points of 9, 12, and 15 months. Okay, but however, we also looked at whether, you see the numbers that we see here, are they proteins which are similar or they are different proteins, which leads us to the next results here. Okay, so for our mass spec analysis just now, in total, 347 proteins were identified using the Max1 software. So when expanded further, okay, this figure actually shows you the similarities and differences between protein identified in human milk for all time point. Okay, the middle part here you see 120, that is which is about 35% of the proteins are actually found present at every time point. Okay, for the outer layer of the Venn diagram here, these are protein detected at specific time points. So you can see quite interestingly that there are quite a number of proteins that is found differentially at the specific time points. Okay, so for the current at the present time, our analysis is focused for the proteins, 120 proteins that are found at every time point. Okay, so next, actually, we did sort of like for the 347 proteins identified through the mass spectrometry analysis, we did gene ontology enrichment analysis, whereby the identified proteins were then classified into different components such as cellular components, biological process, and molecular function. Okay, the colored bar here represents the different months of the milk sample, whereas in terms of the length of each bar, it signifies the percentage of proteins that are assigned to their corresponding functions. Okay, and we also, since for the mass spec analysis, we actually perform quantification by label-free method. So we could actually determine protein differences by using a software called Perseus. Okay, so this is the heat map that is generated, and over here you see the red bars and the green bars. So red means high and green means low expression. Okay, so the heat map of this whole protein abundance profiles were then clustered based on Euclidean distance, and we found 72 proteins that were significantly regulated across the 15 months of lactation. Okay, so following that, we actually looked at the trends of expression, and we could flag 11 proteins that show significant changes in their regulation based on sort of like the time series. Yeah, so the 11 proteins were, you can see that in terms of the trend for the upregulated are highlighted here in red lines, and those that are downregulated are the ones which are in green. Okay, and for, let's say, example, for the downregulated protein, which is SIL1 and CHRDL2, you could see that the level of protein actually goes down quite dramatically 6 months onwards. Yeah, so alternatively, the upregulated proteins also had sort of like a similar trend at 6 months onwards, that sort of like the expression level goes up. Yeah, for most of the 11 proteins. Okay, so we delve on further for the 11 proteins that we found significantly expressed for the time series up to 15 months. So these are sort of like the details in terms of the gene names and also specific biological process that the proteins are involved in. Okay, so for the two downregulated protein, okay, in terms of the biological processes, it is involved in cell structure and function. And in terms of the upregulated protein, okay, it is involved in immunity, metabolism, and also cell structure and function. Okay, maybe I can highlight, one of the proteins that are found to be upregulated, which is transcobalamin. Okay, so transcobalamin is actually, in terms of biological process, it's involved in cobalamin and also cobalt ion transport. Okay, so this would be important actually. Transcobalamin, or cobalamin actually binds to vitamin B12 and forms a complex. Okay, and mainly this binding is actually important for vitamin to protect vitamin B12 in the acidic environment of the stomach. Yeah, so vitamin B12 actually plays an important role in maintaining nervous system function and also cell metabolism. So we sort of like hypothesized that this protein was found to be upregulated from the 3rd to the 15th month. Mainly, it could be due to the increase of demand for this protein caused by the, so as I was saying, that the proteins sort of like are upregulated maybe due to the demands as the child grows, and also for the development in terms of nervous systems and also the hematopoietic development. Yeah. Prof. Azlina, are you able to see the full slide right now?
We've lost your slide.
No.
Okay, hold on. Yeah, I'm sorry about that.
All right, that's fine.
Okay, fine. Right. Okay. Okay. So having said that, just now actually I was actually going to summary already, whereby overall the 11 proteins that were significantly regulated that we saw throughout the 15 months are related to immunity, cell structure, function, and metabolism. Okay, all these are actually very pivotal for the infant's development of growth. So based on the results of our analysis, we can see that breastfeeding actually confers benefit to the child, especially in terms of growth. But having said that, this is actually our preliminary data. We hope to complete the collection and analysis of milk samples up to 24 months. And also having said that, remember I said that right now what we've analyzed is just in terms of proteins seen throughout the 15 months of lactation. There are also interest for us to actually look at proteins which are specifically expressed during the different lactation months. Yeah. So moving forward, okay, we hope that this kind of evidence can be presented to encourage mothers and also to motivate mothers to continue breastfeeding for a longer period of time, since now they know that there are a multitude of proteins and other factors that can actually help in terms of the development of physical development and also cognitive development of their child, and also in terms of protection towards their health in a longer period of time. Yeah. Okay. So I'd like to also give acknowledgement to we'd like to express our appreciation to milk donors who are willing to cooperate throughout this project. So moms who are willing to actually give us a bit of their precious milk sample. Okay, we truly appreciate your kindness. And this work was supported by a fund from the UMIRG. Okay, so in actual, we are one of the sub-project in the bigger picture of the IRG program. We also have sub-programs with researchers from the Akademi Pengajian Islam and also from the Social Preventive Medicine, FOM, which is Prof Mazda and also Prof Nick. All right. So with that, I thank you, and I'll be happy to answer any questions.
Thank you very much, Puteri, for the very insightful sharing of your research. Certainly, it's very important research. Perhaps we take one or two questions. I will read out to you. One question, Prof. Tuputara is asking, is the composition of human milk affected by preservation, such as freezing? So if you have to freeze the milk.
Right. Okay. So for our research, actually, yes, we are aware in terms of there may be fluctuations in terms of the content of human milk, such as freezing, as suggested just now. So due to sort of like, our focus is actually for the protein content of milk. We are very careful in terms of from the point of collection, when mothers actually agree to donate milk, what we usually do is really explain to them how to go about in terms of storing the sample for us. Okay, so when we receive it, it's usually in sort of like a frozen state already, and we store it straight away in our freezer at -20. So only when we are ready to actually extract the samples, okay, then we would actually thaw out. And at that point of time, we will do, actually, when I mentioned just now, is just of extraction for protein analysis, but in actual, we also at the same point of time do extraction for lipids and also for metabolites. Yeah, so reason being that we do not want to have samples that are free stored too many times, then that might affect sort of like the results we obtain, or sort of like what we see may not be what is actually happening in the biological sample.
All right, thank you very much. Maybe one more question. Prof. Chan is asking, why can artificial milk be made to be more like human milk as much as possible?
Okay.
Now, increased fats in formula normally cause chubbiness in
yeah, right. Okay, so I know about that. Okay, if even right now, if you're talking about sort of like other type of milk that is being sort of like everyone is really trying to make milk or formula milk, artificial milk, as close as possible for to humans' milk. Yeah, and it's not just formula milk. Okay, when they come up with different formulations where you can see sort of like suddenly you have milk with MFGM, okay, whereby previously they did not add these factors in. MFGM stands for milk fat globule membrane, because manufacturers suddenly realized that, oh, these are important components for child's brain development. Okay, and if that's formula milk, but there's also milk that is sort of like being cultured right now, okay, in labs, trying to mimic as close as possible sort of like the content of human milk. Okay, maybe in terms of content, in terms of nutrient and the bioactive factors, may be very similar already. Okay, I sort of like read the article, but I believe there's still other factors that may not be able to be given when you give this sort of like artificial cultured milk, you know, such as the bonding with the mother. Okay, that is actually at the time where mothers actually spend a lot of focus on their child, and it's not just giving nutrient, but it's also giving love and care. And during that time, when you talk to the babies and things like that, that is also the process of sort of like brain development for the child. So yeah, I think maybe one day, you know, like scientists or manufacturers may be able to mimic human milk, but nothing beats sort of like the mother's love and care.
Thank you very much, Puteri. So for the interest of, there are several other questions, but for the interest of time, perhaps you can answer the questions by typing in the chat box.
Okay, I will do that. Right.
Okay, thank you very much, Puteri, for very insightful sharing, and I wish you all the best in your research.
Thank you.
And I end my session and I pass to Prof. Kiew. Thank you.
Thank you.
Thank you, Prof. Azlina. Good morning, everyone. I'm Kiew from the Department of Pharmacology. So it is my pleasure today to introduce to you our second speaker. He is Associate Professor Dr. Tan Choo Hock from our department. Dr. Tan pursued his MBBS degree at our university, the University of Malaya. He then completed his PhD and also Doctor of Medicine, focusing on the research into pharmacology, toxicology, and the omics of the venom and antivenoms. He is currently serving also as a member of the WHO Snakebite and Envenoming roster of experts. So today he'll be sharing with us the issue on snake bites and envenoming, which is a much neglected topical disease, and some of his recent research findings in solving the challenges in the treatment of snake bites. So Dr. Tan, the floor is yours.
Hi, can you hear me now?
Now we can hear you.
Yeah, okay. Sorry for the internet. Anyway, thank you for having me on. Thank you for the very kind introduction, Dr. Kiew. So I have prepared a slide, and in order to ensure smooth running of the slides, I have prepared the slides. Hi, good morning. Thank you for... So I'm going to share with you this talk on the title Snakebite Envenoming as a neglected tropical disease, and some of the findings, how we our research may serve to solve these challenging problems. Hi, good morning. Thank you for the introduction and the opportunity to share with you the talk with the title Snakebite Envenoming as a neglected tropical disease on the unresolved challenges and new opportunities. Snakebite envenoming is a multi-organ disease caused by the toxic effect of snake venoms. Each year, around 81,000 to 138,000 people die because of snakebite envenoming, and about 400,000 survivors actually end up with permanent disabilities due to amputations and other crippling disabilities. The World Health Organization recognized this condition as a neglected tropical disease. And WHO has come up with an envenoming roadmap that tries to address this long neglected public health issue by looking at the various strategies. So one of the pillars of the strategies is to ensure the availability of safe and effective treatment, which is essentially dealing with antivenom. But there are challenges that we need to acknowledge in terms of the supply, the efficacy, and also the safety of antivenom products. So speaking about venomous snakes, we got to remember that there are diverse venomous snake species. There are more than 600 species, of which at least half of these are highly medically important, and they are widely distributed in many continents. So when it comes to antivenom treatment, which is rather species specific, then we run into the problem of having limited species coverage and also antivenom production and supply. So from our perspective, we asked just one question: how can we improve the treatment for snakebite and venoming? And these are our thoughts. The snake venom compositions, they are so variable at both interspecies and also intraspecies levels. And the variation in the key toxins actually results in these differences in the toxicity, and also is accompanied with the variable antigenicity of the venom. Therefore, deciphering the variation is very crucial in order to understand the pathophysiology and to improve the antivenom treatment. And lastly, besides antivenoms, there should be other innovations to be developed for new treatments against the snakebite and venoming. So our research begins from the field to bench, where we characterize the snake venoms and we look at the genes, in particular the venom genes and the toxin genes related to the various diverse species across many regions. The basic knowledge is then applied, from bench to bedside, where we look at the diagnostics and also the treatment, with the hope that the outcome will be able to delay manifestation of toxicity so that the patients can buy time to reach healthcare facility for proper treatment. And then the knowledge should be also able to improve pre-hospital management and first aid. Then with rapid and accurate diagnosis, it is hoped that the proper treatment can be given to patients. And lastly, is very related to our basic research, is on the improvement of antivenom quality in terms of its purity and efficacy. So the venomic research, or I would say functional venomics, that we are practicing is one that is rather interdisciplinary, transdisciplinary. So in the beginning, we look at the biochemical composition of various snake venoms through methods like proteomics and transcriptomics, and then we move on to the functional level in terms of the toxicology and pharmacology, and this is where various in vitro and in vivo assays and models are established. Then we look at the immunological aspects of not only the venoms but also the antivenoms, so that we can recognize or understand the strengths and also weaknesses of various treatments available. And also we have an idea of how the different antivenoms vary from one to another in terms of their neutralization profiles. Then having this knowledge, this will enable us to move on to the innovation of antivenom. And what we are currently working on is to come up with a certain types of so-called novel antivenoms and also other emergent new therapies to mitigate the toxicity of snake venoms, so that the treatment of envenoming can be improved. So I would like to illustrate the research using a series of cobra venom studies. You can see this is a cobra, but bear in mind cobras are much more diverse than this. And in Asia alone, there are more than 10 species actually, and many other subspecies of cobras, and they are distributed in various localities across Asia. And if we look into the venoms, they are quite variable in their toxin components or compositions, but invariably they contain two similar groups of key toxins, what we call the alpha neurotoxins. And these two forms are various proteoforms of short alpha neurotoxin and also long alpha neurotoxins, which act postsynaptically and blocking the neuromuscular transmission, much like the curare toxins that you know from pharmacology. And the abundance of these alpha neurotoxins in each venom is well correlated with the LD50 or the lethality of the particular cobra venom. And this explains the variability in lethality of the different cobra venoms across various geographical areas. Of course, we also need to know that the snake venoms, the variation is not just only in this lethality. The variation often comes with differences in protein antigenicity and also the neutralization profiles. So to make situation more complicated is that the treatments for various cobras, for instance, cobra venoms vary from region to region, and the quality, the efficacy, the purity of antivenoms products available in the different regions also vary from one to another. So our work, basically, we conducted the immunorecognition and also neutralization study against the many cobra species that we found across Asia and also some part of Africa. And we use basically most of the antivenoms that are available in the regions. We test the homospecies or the homologous neutralization, and we also test the paraspecific cross-neutralization capacity of the different venom against the different antivenoms. So this is one of the assays that we commonly conducted on a nerve muscle preparation. This is to test for cobra venom neurotoxicity. This is an example where we actually tested the Thai cobra antivenom, looking at the potency of neutralization against the same species, what we call the monocle cobra, from 3 different areas in Malaysia, Vietnam, and also Thailand. So we found that the Thai antivenom itself actually requires 4 times higher doses to prevent paralysis caused by this particular species originated from Thailand. And this is actually consistent with the presence of a much higher abundance of the alpha neurotoxins in the Thai species of the monocle cobra compared to that in Vietnam and also Malaysia, and therefore indicated that the abundance of these alpha neurotoxins is actually a determinant that will affect or influence the dosage management. So this is validated in vivo study, where in the in vivo neutralization study, we found that the efficacy of the antivenom is against the lowest, or the neutralization is the weakest against venom that contains much higher alpha neurotoxins, such as in this case is a Thai cobra species. So the findings together indicates that the higher the alpha neurotoxin abundances in a venom, then that is almost like a predictor that the antivenom efficacy is going to be low. And clinically, this means that the higher dose of antivenoms, either given in a much higher dose, a bolus dose, or you have got to give repeated doses, will be required to save the patient's life or to prevent paralysis. But clinically, unfortunately, this also means that the patient is predisposed to a higher risk of hypersensitivity reactions, which is equally fatal and also incur higher costs. We also examine the effect of time of treatment in reversing the muscle paralysis caused by snake venoms. So we consistently observe that in cobra envenoming, the later the antivenom is given, then from the time of paralysis onset, then it becomes even more difficult to restore the nerve muscle functionality. So this infers that in cobra blood envenoming, antivenom should be given or commenced early as possible once it is indicated, in order to achieve a better prognosis and also a better clinical outcome. Then we also established the experimental envenomation model, or what we call the challenge rescue model, where the animal is experimentally inoculated with venom, mimicking the real-life envenoming situations, and then the antivenom is subsequently given as indicated by the onset of paralysis, much like clinical practice. And we observed that in cobra venoms that contain or that is rich in alpha neurotoxins, then it is associated with very rapid onset of neurotoxicity. And in this kind of envenoming, then typically very high doses of antivenom is required, and sometimes even require multiple dosing. And it also takes a rather long recovery time for the animals to recover from paralysis and weakness. So this indicates that the cobra antivenoms, basically all the cobra antivenoms available, has limitations in terms of the potency and also the abilities to neutralize the neurotoxicity caused by the various cobra species. And our work doesn't stop at cobras. We further extend the characterizations to various other highly neurotoxic snakes in the regions from the land to the sea. So this is important because we want to actually look at the variations in terms of the venom compositions, the key neurotoxin contents, and also the variations in terms of their immunology and also the neutralization profiles in response to treatment of antivenoms. So with the pharmacological characterization and also the Venomics protocols that we have established in the lab, we are able to identify and also purify the various key toxins from the diverse venomous species in the region. And this actually opens the door to many exciting works, which include the development of diagnostics, and then the application of toxins in the field of drug discovery. And most importantly, where this talk is relevant, is the development of next generation antivenom. So based on our findings from the pharmacological characterization and also the Venomics guided toxin purification, we came up with a principle for antivenom improvement. That is to get the right target toxins for the right species tailored to its locality. We innovated an immunogen mixture which contains basically the diverse key toxins from the various species across the region. And we use this immunogen to hyperimmunize horses in collaboration with our friends from Thailand. And this pilot antivenom appears to be so strong or powerful that it is able to cross-neutralize many neurotoxic venoms, almost 3 dozen of them from across 4 continents. So this is an exciting finding, but of course it is still at the laboratory or preliminary stage, and my team is currently looking into possible refinements of the product, where we're looking into the refinements of the antigen formulation, and then the improvement of the immunizing protocols, and also the process of antibody purification, with the aim to achieve a wider spectrum of species coverage, and then to achieve a higher efficacy and potency, and also a better purity and safety profile of the antivenom product. And at this point, let's take a look back at the Venomics and try to understand its impact or its contributions to the field of snakebite envenoming as a neglected tropical disease. Now, Venomics is, you can see now, is basically a platform, an integrated platform of proteomics, transcriptomics, and genomics, supported by the high throughput advancing sequencing techniques, be it for proteins or RNA or DNA, then along with the advancement in bioinformatics and also the ever-growing databases. So Venomics contributes to various aspects. For what we are concerned, of course, is from the medical perspective, where there is possibility for bioprospecting and then potential for drug discovery. And then for the treatment aspects, then it is possible to improve our understanding of the pathophysiology of envenoming, and the knowledge about snake venom complexity and diversity, and therefore the possibility of improving treatment in terms of antivenoms. But the treatment management doesn't stop at antivenom alone, because there should be also other modalities besides antivenom to tackle or to handle, to overcome the issue of snakebite envenoming. So if we look at the Venomics findings, this is based on proteomics and also the transcriptomics that we have got for the various species, major species of cobras across Asia. Of course, there are also those from Africa, which I did not include here. But you see that sometimes, from the evolutionary point of view, you can see the dichotomous venom phenotypes in this case, where you see there is a shift from the long neurotoxin dominant phenotype to the short neurotoxin dominant phenotypes, from the West to the East Asia. Of course, the implications is much complicated, but to make it simple, I would say that the variation and the change in this venom phenotypes, of course, it happens at the genes level, but this is definitely going to bring a clinical impact, because the change in the antigenicity of the toxins, and also the clinical manifestation of and the severity of the toxicity is going to vary from species to species and from region to region. So that we found that in snake venom study, it is not sufficient to only reach the level of snake venom and the genes. What I meant is we got to delve even deeper into the the very genes of the very, heavy toxins and the venoms pertaining to a particular species. And so I think the concept is we used to say, from the field to the bench. And in the field, what we used to do is we identify the snakes, we authenticate the specimens, and then we collect the venoms, and we're happy with it. But it should not just stop at the level of venoms or the fangs. In fact, we should go even all the way back or all the way into the snakes and look at the variations and the complexity of the toxins at the genetic level. And this is where genomics comes in. So the snake venom genomics, it aims to unveil all the genes in the body and also in the venom production. And of course, it should be able to provide deeper insights into the evolution of the snakes, of the venoms. And importantly, you get a lot of information about the structures and the functions of snake venom toxins from the genomes. And this is it going to, be beneficial for our, medical practice and medical application? Of course it is, because it's going to be helpful for next generation antivenom development and potentially antidote development. So people are talking about synthetic monoclonal antivenoms, so that can be done, in light of genomic information and the targeted toxins of various species, like what we have been doing, using the earlier platform of phenomics. So that can be even consolidated to create a pan-region, a pan-species antivenom that can be useful and available for many populations across the globe. Then lastly, of course, by looking into the genes of the venoms, then it is possible to identify new potential targets for the development of adjunct treatment. So if you take a look at the venom proteomes of most cobras, and in this case is the Philippine spitting cobras, you will see the dominance of 3 finger toxins, which contain variable amounts and a diversity of alpha neurotoxins. But there is always also the presence of what we call high molecular weight toxins within the venoms at a much lower abundances, typically like less than 1% of total venom proteins. So in this case, I gave an example of the presence of phosphodiesterase, which is, the phosphodiesterase is much like the EMPP in the body proteins, which is a transmembrane regulatory proteins. But why is it, ended up in snake venoms and becoming a secretory proteins in the venoms? And it's huge, like, almost 100 kilodalton. So the passing question here is why the evolution in the venom keep these proteins? So these are apparently some passing questions that you will require some depth of genomic knowledge in order to figure out or to find out the answer behind it. So for the evolution to keep these expensive proteins in the venom, we hypothesize that there must be a function. Invariably, this will be related to some form of predation, diet, or even defense in order to ensure fitness and survival of the species. So if you look at the activities then of these toxins, then it actually would be sort of contributing to the overall venom activity or toxicity, or it put in another word, that would be envenoming. So if we are able to inhibit these high molecular weight toxins, in a way, it may be possible to actually mitigate the toxicity or even, prevent or reduce the spread of the venom before the toxicity becomes a full-bloom. So therefore, it would be possible then to reduce the dependence on antivenom treatment. So following the genomic study and the structural study of the high molecular weight toxins, etc., which is in collaboration with our friends in Taiwan, then we proceed with the functional study, and we actually found that the use of certain inhibitors, in fact, are potentially therapeutic because they actually reduce the toxicity of the venoms and improves the survival without antivenom. So that basically means, these inhibitors can be applied and even preclinically or at the pre-hospital setting and without the dependence of antivenom. So this will greatly improve the overall management of snakebite envenoming at the various levels in the future. Just recently, we demonstrated that the use of farasplatid, which is a small molecule inhibitor of secretory phospholipase A2, was able to mitigate the toxicity of various acyclic clade venoms, which are essentially these phospholipase A2 from clade venoms are presynaptic neurotoxins. So by inhibiting these toxins with farasplatid, we see that it is possible to reduce the lethality and also to improve the survival time and the survival rate of the mice envenomed with the clade venoms. So this is rather exciting because it shows that the use of the right proper inhibitor against the key toxins, even in the absence of antivenom, can be beneficial for the improvement of snakebite envenoming treatment in the future. So before I end my talk, I would like to acknowledge and say thank you to all my collaborators from all these institutions, and also my students and my collaborators in and outside of University of Malaya. Without you, this work cannot be completed. Thank you.
Thank you, Dr. Tan. So now we go on for the Q&A session. So the first set of questions are from Prof. Chan of anesthesiology. So the first question, snake kills in many different ways. Rapid threat of death comes from impact on the CNS. How can we ensure drugs are administered physically early enough to have good damage control? Dr. Tan?
Hi, thank you for the questions, Prof. Basically the venoms, of course, the different venoms from different species, the onset in general are different. So the one that you mentioned about rapid onset of death, especially usually affects the neuromuscular junctions, and then that leads to respiratory failure, and without oxygen, you die very fast. So the physically enough is always a challenge. So what we can propose is, which leads to another question, is that the preclinical management, the first aid particularly, should be optimized, and this needs a lot of field work, bringing the awareness, proper first aid, aiming to reduce the spread, because the venom basically they spread by lymphatic system. So if you can slow it down, then you can buy time to reach the hospital for administration of antivenom. On the other hand, the hematological system effect, yes, as you say, usually the onset is slightly slower. So patients may be able to reach the hospital in time. But there's the ideal situations in the rural. Sometimes patients have to travel, several days to reach hospital, in some countries. So there is always a challenge. So apart from the antivenom administration, we actually also advocate the proper and appropriate pre-hospital management, which include the correct first aid.
Okay, thank you, Dr. Tan. Second question from Prof. Chan as well. Must all antivenom come from biological source? Can they not be manufactured like the way insulin or other hormones can be manufactured?
Yeah, I mentioned a little bit also about the synthetic antivenoms. So there are various strategies or techniques that are being tested at the moment, which includes recombinant antivenoms, etc., humanized antivenoms, and so on. But the things about, if you compare it to synthetic insulin, for example, the snake venom toxins are much more diverse. There are so many variations and then so many types of toxins, and within even each species from different regions, they have variations. And the antivenom basically works at the level of antigen or epitopes recognizing. So it remains a very far-fetched, I would say, but it is a possible solution. But of course, there's always issues about cost and so on. When you are trying to market this in those poor and impoverished populations, then this kind of fancy, expensive synthetic antibodies will become always an issue. So the biological ones is still the current antibodies, the current treatment.
Okay.
Thank you.
Okay. So the next question, will you comment on this? Maybe antivenom should be present at the site where they are required, for example, estates where snakes are abundant. Is this possible in current situation?
Yes. The thing about antivenom is it has to be administered by trained, usually doctors, because the way to administer it is by intravenous, infusion. And of course, you also need to establish the need of antivenom before you administer it. So the practice is you need to administer it under supervised medical care. Making it available in estates is a challenge because, the administration issue and so on. But also, we would rather that, a proper awareness is being raised in these populations, those, in the estates, for example, that when they are bitten, they do the proper first aid with the aim to slow down the spread, minimize the complications, and then as soon as where possible, go to the nearest medical facility. And this, of course, also have to address. We need to address also sometimes, the influence or from the so-called traditional practitioners, which actually delay the treatment seeking.
Okay. Quickly, the next question: does antivenom works on tissue damage?
Okay, I was trying to type the answer, but anyway, that the tissue damage, I think you refer to local tissue damage such as necrosis. Yes, studies have actually found that the role or the effectiveness of antivenom against local effect is much less, compared to the systemic one. Basically, probably is to do with the biodistribution of the antivenom to the local sites, the skin and so on. So it's not so, I would say, but perhaps not so effective clinically. But there are other, treatment. That's why we are looking into the use of, for example, inhibitors and also other modalities, like first aid and so on, to minimize the local effect.
Okay, due to the interest of time, this will be the last question from Prof. Tunku Sarah. Is there a possibility of immunization against the venom?
Okay, this has been tried, I think, 30 or 40 years ago. But the question is again back to the diversity and the variability of snake venoms. So you can't be possibly, immunized to get the very effective antititer that covers the variations in of the different toxins in the different snakes. So at the moment, this is actually not in the in the options. Theoretically, you can do it, but it is not. It's unlikely going to work.
Okay, thank you, Dr. Tan. Due to the interest of time, we have to end the session now. So we will answer the rest of the questions in written form later. Okay, thank you everyone for attending. See you in the next round. Bye.
Thank you very much. Bye.